Hello everyone. We are excited to introduce AMPDesign, our new tool that is part of the latest version of our DBAMP database. AMPDesign is aiming to create antimicrobial peptides, also known as AMPs, that target specific bacterial species such as E. coli and Staphylococcus aureus. The AMPDesign tool is built on three key components: generative adversarial networks, reinforced learning, and our previously published AMP activity predictor. The diagram on the left illustrates how AMPs are generated using generative adversarial networks. Our training set is entirely sourced from DBAMP database. Generative adversarial networks consist of two parts: the generator and the discriminator. The generator continuously creates new sequences and submits them to the discriminator for evolution. The generator and discriminator iteratively improve through this process until the discriminator can no longer distinguish between the sequences created by generator and real ones. The image on the right is a schematic of our reinforced learning strategies. Beginning with the first amino acid, the sequences are developed step by step using the Monte Carlo method. Each potential amino acid sequence generated through this method is then evaluated by both discriminator and the activity predictor. Here, let's revisit an important concept in our reinforced learning strategy, the reward function. The reward function combines the outputs of the discriminator and the activity predictor. Our activity predictor, a deep forest-based tool we previously published in Protein Science, plays a crucial role. It helps AMPDesign generate highly active potential AMPs by leveraging reinforcement learning strategies. Our experimental results demonstrate that AMPDesign effectively generates highly active AMPs targeting specific bacterial species. For instance, we used AMPDesign to generate 100 potential AMPs against E. coli. Our activity predictor revealed that 82 out of these 100 sequences displayed high activity towards E. coli. We further analyzed the common peptide descriptors and features of these sequences using the UMAP, the dimensionality reduction method. This allowed us to observe the distribution of both the training set sequences and the newly generated sequences in hidden space. In Figure A on the left, we can see that the distribution of the generated sequences and the original sequences are very similar. This similarity indicates that AMPDesign effectively learns the characteristics of peptide sequence patterns, enabling it to generate corresponding AMPs accurately. Figures B through I display the physical and chemical properties of both the original and generated AMPs. The distributions of these properties are also very similar, suggesting that the newly generated 82 AMPs are highly likely to inhibit E. coli. This inhibitory effect is due to their physical and chemical properties closely matching those in our dataset. We have named these 82 potential peptides DBAMP G0001 to DBAMP G00A2 for further research by users. Based on previous literature indicating that lipopolysaccharides are targets in gram-negative bacteria, we conduct a docking study. We randomly selected DBAMP 06098, an anti-E. coli sequence from DBAMP, as a reference sequence and docked it with lipopolysaccharides. Additionally, we randomly selected 5 sequences from our newly generated 82 sequences and docked them with lipopolysaccharides as well. The docking results of the new sequences closely match those of the reference sequences. These further confirm the advantages of AMPDesign in generating high-activity AMPs. AMPDesign is available for free on our GitHub repository. We've added a section called Generation, which you can find on our DBAMP homepage. If you have any question while using AMPDesign, feel free to email us. Our team will respond within 2 days. Thank you for watching this video to the end.